Hello friends and welcome to my channel Easy Life Science. Here I am talking about the important questions, their answers with detailed explanation for set exam subject Life Science part 11. So please subscribe my channel Easy Life Science, like my video, share it to your friends and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Question first. In Whittaker's five kingdom system, all primitive eukaryotes were bunched into the group. The number of scientists used a number of classification system for organisms. The five kingdom system of classification was proposed by R.H. Whittaker in 1969. The five kingdom system formed on the basis of characteristics such as cell structure, mode of nutrition, source of nutrition and body organization, and the five kingdoms given by a scientist Whittaker were kingdom Monera, Protista, Fungi, Plantae and Animalia. These were the five kingdoms given by a scientist R.H. Whittaker. And the characteristics studied for this classification were cell organization, presence or absence of cell wall or its composition, nutrition, mode of nutrition. So let's look on this image. The kingdom Monera. In a kingdom Monera, R.H. Whittaker placed all unicellular organisms. In a Protista also the unicellular organisms. The kingdom Fungi, in this unicellular and multicellular organisms were kept. In the kingdom Plantae and Animalia, multicellular organisms were kept. The cell wall Monera, the cell wall of a organism present in a kingdom Monera made up of a peptidoglycan. The Protista, these were the organism which lacks a cell wall, but some organism in the Protista shows a presence of cell wall. The cell wall of a organism present in a kingdom Fungi were made up of a chitin. That of the Plantae, cell wall is made up of cellulose, and a kingdom Animalia, no cell wall present in these animals. Nutritional class, the kingdom Monera, some of them are autotrophic and most of them were heterotrophic. For Protista, some of them are autotrophic and most of them were heterotrophic. Fungi, all of them were heterotrophic. And kingdom Plantae is the only kingdom from which the organisms were autotrophic. That means they synthesize their own food. And a kingdom Animalia, in this class, in this domain, the organisms were heterotrophic in nature. They are depends on the autotrophic organism for food. Mode of nutrition in Monera, absorption. In case of Protista, it by ingestion or absorption. In Fungi, by absorption mean. In Plantae, absorption, and in Animalia by ingestion. Example, the bacteria or a true bacteria, archaeobacteria and cyanobacteria are placed in kingdom Monera by a scientist R.H. Whittaker. In a Protista, algae and protozoa were kept. In domain Fungi, yeast, molds and mushrooms. In kingdom Plantae, mosses, ferns and flowers. And in kingdom Animalia, insect, birds, fish and human beings. So this is a classification given by a scientist R.H. Whittaker, and these were the five kingdoms. Also the three domains of a life like archaea, bacteria and eukarya, and these three domains system of classification introduced by a Carl Woese. Three kingdom system given by Ernst Haeckel, Animalia, Plantae, and Protista. So these are some attempts made by a different scientists for a classification of organism. But here in this question, in Whitaker 5 system of classification, all primitive eukaryotes were bunched into the kingdom Protista. So correct answer of this question is option second. Then the next question, microscopic unicellular organism characterized with food gullet, flagellum, chloroplast, mitochondria, and a nucleus. Can it qualify itself the member of which kingdom? So these all characteristics, the microscopic unicellular organism, a nucleus, mitochondria, and chloroplast. This is a characteristic of organism from kingdom Protista. So answer first is the answer of question number second. Then moving to the next question. Also a two kingdom system of classification given by a scientist Linnaeus in 1758, and the two kingdoms were plants and animals. In a plant kingdom, all the photosynthetic organisms were kept, and in animals, non-photosynthetic organisms were kept. Two kingdom system by a Linnaeus, three kingdom system by Ernst Haeckel, and three domain by Carl Woese, and five kingdom by Whitaker. So the next question, examine the species listed and recognize that which belongs to a Indian center among 8 center of origin recognized by a scientist Pavlov in his attempts to study the domestication of a crop. This is a question asked about the geographical plants or its center, and that of the present in Indian center. A center of origin is a geographical area where a group of organisms, either domesticated or wild, first developed its distinctive property. They are also considered as a center of diversity. Centers of origin were first identified in 1924 by a scientist Nikolai Pavlov, and 8 centers were there, including an Indian center. And here question is asked on Indian center, and the species which listed amongst are from Indian center. For this, the option second, cluster bean, pigeon bean, orange, lemon, and sugarcane, these all species or these all plants are from Indian center. So these are the some species listed here from Indian center, which are center of origin, cereals and legumes like chicken pea, pigeon pea, bean, mung bean, rice bean, and a lot of mango, citron, tamarind, cucumber, banana. You can take a screenshot of it, because next time question will be asked on any other from it, and asked which of the following is a species from Indian center. Then next question, two of the Indian spots or two of the Indian hotspots out of 25 identified and declared world over. Two hotspots, that is Eastern Himalaya and Western Ghats. So option second is the answer of this question. Then next question, two sympatric species going in a same area must have different character where as two allopatric species growing in a different geographical area may have a similar morphological characteristics, which is known as a character displacement. Character displacement is the term used to describe an evolutionary change that occurs when two similar species inhabit the same environment. Under such conditions, natural selection favors divergence in the characters like morphology, ecology, behavior, or physiology of the organism. The two sympatric species means the two species are there, species A and species B. These are living in a same area and not sexually reproducing. But in allopatric speciation, one species get divided from original one and change its character, and its habitat also get changed because of a physiological barrier. This is known as allopatric speciation, and these two species A and A dash does not reproduce. So this is known as a character displacement. The next question, which of the following is the best description of the term ecosystem? The ecosystem means a geographical area where plants, animal and other organisms, weather and landscape work together. Ecosystem contains biotic or living parts as well as abiotic or non-living parts. The biotic parts include plants, animal and other organisms. The non-biotic plant includes temperature, humidity and osmotic pressure. The types of ecosystem are forest ecosystem, grassland ecosystem, tundra ecosystem and desert ecosystem. And this word ecosystem was first time coined by A.G. Tansley in 1935. So the ecosystem means a community of organisms together with the environment in which they live. Option third is the answer of this question. Then choose the correct set of the eras during Precambrian era. The Precambrian era was started from formation of Earth, that was a Hadean era, then a Archean, and then a Proterozoic era. So this is a sequence. So option second is the answer of this question. Then next question, all of the following are the parts of the allopatric speciation by a natural selection except. The allopatric speciation means one species moved to another environment and the second environment get physically separated by a barrier like mountains or a river, and it get changed in its morphology and leads to a speciation. So after speciation, the changing of a species or the changed species does not mate with original species. So it is known as allopatric speciation. So read one by one. A geographically isolated population evolves or change in response to a novel environment. It is true for allopatric speciation. Reproductive isolation evolves as a byproduct of changes in other traits associated with adapting to a new resource or environments. It is also true about ecosystem. Selection against hybridization leads to a exaggeration of signals to facilitate recognition of corn species. So it is not true about allopatric speciation. Changes among or between population occur while a population are geographically separated. It is also correct. So third option is the answer of this question. The next question, find the wrong cause of a sympatric speciation. Here asked about a wrong cause of a sympatric speciation. Adaptation and a differentiation within a population can lead to a speciation. Yes, it is true. Evolution of a distinct population or a variety via mimicry. Yes. speciation among insect can be initiated by adaptation to a different host plant as a source of food. Yes, it is also true. Population divided by a budding of a small, completely isolated founder colony form a larger population. This is not true. This is not for a sympatric speciation. So option 4 is the answer of this question. Then next question, enzymes are required for making a plant protoplast. The plant protoplast, this term is coined by a scientist Hanstein. Protoplast means a cell without cell wall. Protoplast can be generated by a stripping the cell wall from a plant, bacteria and a fungi by a mechanical, chemical or a enzymatic means. Protoplast differs from a puroplast. Protoplasts are used for a DNA transformation. So here the question is asked for enzymes that are required for making a plant protoplast. So answer of this question is a cellulase and a pectinase. Not one. The answer of this question is option second. Here type of cell and enzymes required for a preparation of a specific protoplast. For plant cell protoplast, enzymes required are cellulase, pectinase and xylanase. For a protoplast preparation of a gram-positive bacteria, enzyme like lysozyme, diacylmuramidase and lysostaphin. These three are the enzymes required for a preparation of a protoplast of gram-positive bacteria. Means by using lysozyme and diacylmuramidase enzyme and lysostaphin protein, the gram-positive cell wall get degraded. So we can prepare the protoplast of it. The enzyme chitinase used for a protoplast preparation of a fungal cell. So for this question, enzymes required for making a plant protoplast are the cellulase and a pectinase. Not one. Option second is the answer of this question. Then the next question, hybridoma is formed by a fusion of. Hybridoma technology is one of the most common method used to produce monoclonal antibody. Monoclonal means which are identical. In this process, antibody producing B lymphocytes are isolated from the mice. After immunizing the mice with a specific antigen and fused with immortal myeloma cell lines to form a hybrid cells, and this technique is known as a hybridoma cell line formation technique, and this technique given by Georges Kohler and Cesar Milstein. So here question is asked, hybridoma is formed by a fusion of, it is formed by a fusion of plasma cell and a myeloma cell. Humans differs from one another by how much? They are differs on an average 1 nucleotides in 1000. So option second is the answer of this question. In affinity chromatography, the required ligand is often released by. Affinity chromatography separates the proteins on the basis of interaction between a protein and a specific ligand. The binding of the proteins to ligand which are attached to a matrix is reversed by either competition or by decreasing the affinity with the pH or ionic strength. The pH of a solution determines the physical states of the protein contained within based upon the pKa values of their amino acids. Different proteins have different ranges in which they are stable or will bind to other proteins. So, in affinity chromatography, required ligand is often released by. It is released by changing the pH. Option first is the answer of this question. And next, protein A, which has a strong affinity to a Fc region of immunoglobulin, is extracted from. Protein A is a 42 kilodalton protein. It is a surface protein found in the cell wall of bacteria, that is a Staphylococcus aureus. It is encoded by a SPA gene, and its regulation is controlled by a DNA topology, cellular osmolarity, and two component system. It has found to use in a biochemical research because of its ability to bind immunoglobulins. It is composed of 5 homologous immunoglobulin binding domains that folds into a 3 helix bundle. It binds to a heavy chains within a FC region of the most immunoglobulins. So here the question is asked, the protein A is extracted from, it is extracted from the organism Staphylococcus aureus, and this protein has a strong affinity to bind with a FC portion of a immunoglobulin. Then moving to the next question, which of the following microscopic technique relies on the specimen interfering with the wavelength of light to produce a high contrast image without the need for dyes or damage to a sample? Here in this question, the question is asked on a microscopy and observation of a slide without any staining or by using any dyes or without damaging the sample. So one technique is there where there is no use of dyes, that is a phase contrast microscopy. So this is the answer for this question. So thank you for watching me. If you like my video, please subscribe my channel Easy Life Science, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.